students, our first lecture is about cell structure. By the end of this lecture, you would be able to define the cell, something you have already done in O levels. The next topic is a bit new, that is to differentiate between prokaryotic and eukaryotic cells. Then we will go through different type of microscopes and how we are able to analyze and identify different structures of plant and animal cell under both light and electron. Okay, what happened was that initially Robert Hooke, the scientist, studied different slices of cork. Cork is a plant material, and he observed some empty boxes in them. I have given the diagram on the side. He named them cells, and then there were two other scientists who came up with cell theory, and the basic definition that we have today of cells, that is, it is the basic structural and functional unit of all living organisms. Then came another scientist, Frischhaus, who stated that all the existing cells came from the pre-existing cells by cell division. Okay, let's quickly go through the different type of cells on the basis of complexity. Generally, eukaryotic cells are more considered to be more complex than prokaryotic cells, and it is also believed that all the eukaryotic cells came from prokaryotic cells, the basic cells. Pro means before, karyon means nucleus. So prokaryotic cells are those cells which do not have a true nucleus. On the other hand, eukaryotic cells are cells that have the true nucleus. Now, what do you mean by true nucleus is that their genetic material is bounded by a membrane which is also referred as nuclear membrane or nuclear envelope, because of the fact that these are made of two membranes. Prokaryotic cells have poor membrane system. On the other hand, eukaryotic cells have well-developed membrane systems. Example of prokaryotic cells include bacteria, the structure of which you have already gone through in your O levels. Next is cells of an algae, cyanobacterium. The example of eukaryotic cells are animal, plant and fungus, and there are some other members of a group called protoctists. Some of the members of this group are also eukaryotic cells. Okay, now we have to differentiate it on the basis of membrane system. Prokaryotic cells, as I already told you, do not have genetic material bounded by a membrane. Rather, their nucleic acid or genetic material is freely dispersed in the cytoplasm. There are no membrane bounded organelles also. On the other hand, if you see eukaryotic cells, they have a very well defined double membrane around genetic material, forming a very true nucleus. Plus they have organelles that can be singly membrane bounded, or they can have a double membrane or not at all. Organelles with a single membrane are lysosomes, Golgi apparatus or Golgi bodies, vacuole. Organelles with double membrane include nucleus, mitochondria and chloroplast, and organelles with no membranes include centrioles, ribosomes and microtubules. Then there is a different of mesosomes. Mesosomes are nothing but just membrane enfoldings which can be seen in prokaryotic cell but not in eukaryotic cells. Okay, this is a very important slide. It tells you about the difference in genetic material between these two cells. In prokaryotic cells, DNA is double-stranded and circular, while on the other hand, eukaryotic cells do have a DNA in double-stranded form, but the structure is not circular, rather it is linear. DNA is lying freely in cytoplasm in prokaryotic cell, as we have discussed in previous slides. Eukaryotic cells have their genetic material bounded by a membrane. Prokaryotic cells have single molecule of their DNA, while in eukaryotic cells there are many molecules. Prokaryotic cells have their DNA entangled with itself, while in eukaryotic cells the DNA is associated with a protein called histone, histone molecules which form a structure we know as chromosomes. Prokaryotic cells DNA is naked. It means it is not associated with any additional structure. There is no coiling as well. Prokaryotic cells have plasmid sometimes, as you have seen in the structure of bacteria in the chapter of biotechnology in your O-level syllabus. These are additional DNA rings in addition to the main DNA loop. Eukaryotic cell, on the other hand, do not contain plasmid. Okay, this slide discusses about the comparison between the cell walls. Prokaryotic cell wall is highly complex, while in eukaryotic cells, the cell wall is simple carbohydrate. Prokaryotic cell, as we have seen in the cell wall of bacteria, it is made up of murine, a peptidoglycan. It is a complex of sugars, amino acids, and fatty acids. On the other hand, if you see the example of cell wall in eukaryotic cell, we see two of them. In plant cell, it is made up of cellulose, while in fungus, it is made up of chitin. Now, there is another term which is called as gram positive and gram negative. This term is used for prokaryotic cell for bacteria. If the cell wall gets stained, it is stainable. We say that the bacteria is gram positive bacteria. If their cell wall is not stainable, then we say that the bacteria is gram negative. We will go through the details of types of bacteria. Then you will get to know that we differentiate different classes of bacteria on the basis whether their cell wall is stainable or not stainable. So if they are stainable, if they can be stained, we will say that this bacteria is gram positive, while if it does not, we will say that it is a gram negative bacteria. Okay, these are some additional and some very specific structure we see in prokaryotic cell. First, go through the structure that are always present in the cell. The prokaryotic cell. These structures are cell wall, cell surface membrane or simply cell membrane, cytoplasm, circular DNA, and ribosome. These structures will always be present in a prokaryotic cell. On the other side, I have written that structures that are sometimes present in a prokaryotic cells means they are not compulsory to be seen or observed in all prokaryotic cell at the same time. The first structure is for locomotion. It is cilia and flagella. The next structure is of pili or pili. These are smaller than cilia and help in sexual reproduction. Then is capsule. Capsule is for additional protection. Plasmid, already discussed that these are additional rings of DNA. If you remember your syllabus from O levels, we have read that plasmid helps a lot in making products in biotechnology. Then there are some foldings of cell membranes, which again we will go through them. Some of these membranes will be adapted to do photosynthesis, and some of these membranes will be adapted to do nitrogen fixation. So all these structures are sometimes present, not always, while on the other hand, cell membrane, cell wall, cytoplasm, circular DNA, and ribosome will always be present. Okay, this is the diagram of a bacterium, and here you are being shown different structures that are sometimes present and the structures that are always present. So if you see cell wall, which is containing a murine or peptidoglycan, it is the cell wall of the bacterium. Then is cell membrane, cytoplasm, circular DNA, which is sometimes referred as chromosomes, and ribosomes. On the other hand, if you see the structures that are additional, that are sometimes present, there is flagellum, which is a hair-like structure, has a whipping movement and helps in locomotion. Capsule, again present sometimes and provides additional protection. In folding of cell membranes, if you see that the cell membrane is making infolds inside the cytoplasm, these membranes can be adapted to carry on nitrogen fixation or can do photosynthesis. Then there are additional rings of DNA called plasmids, pili, which are smaller than flagella, and they are used for attachment purposes during sexual reproduction. Okay, this is the general structure comparison between prokaryotic and eukaryotic cells. First point is about their size. Average diameter of the cell of a prokaryotic cell is about 0.5 to 5 micrometer. On the other hand, eukaryotic cell is quite larger compared to prokaryotic cell. It is in 40 micrometer diameter, and it is about 1000 to 10,000 times in volume compared to prokaryotic cell. So you can see that eukaryotic cell is definitely quite larger than prokaryotic cell. Next point is about ribosomes. We will go through this point again in later topics when we will read about ribosomes individually. Ribosomes are smaller in prokaryotic cells, and their diameter is about 20 nanometer, while in eukaryotic cells they are quite larger and are in 25 nanometer in diameter approximately. Prokaryotic cells examples we have already discussed. These are bacteria and some of the cells from blue green algae, cyanobacterium. Examples of algae are there in eukaryotic cell as well, along with plant, fungus and animals.